Hi all, Dr. Clark here. Today we're going to demo out lab number 10, or the photosynthesis lab. Okay. The photosynthesis lab has four main parts. We're going to look at photosynthetic pigments through paper chromatography. Then we're going to look at leaf microanatomy. So we're going to look at what are the main parts that make up a leaf. We're going to look at imprints of stomata, so we can get an indication of how much transpiration and how much respiration, the taking in of carbon dioxide and release of oxygen, do certain plants do. And then finally we're going to look at the uptake of carbon dioxide by plants during photosynthesis. So real quick, we're just going to go through the lab, and then I'll show you the videos on each one of those pieces. Okay. So if you remember from lecture, photosynthesis is this basic equation. Carbon dioxide molecules coming in, mixed with water. You add some photons, you're going to get glucose molecule, some water, and some oxygen. Remember, oxygen is a waste product, glucose is the product. Alright, so we're going to calculate the RF factor, or the RF number, for chromatography for different types of pigments. Alright, and we're going to go through this procedure, and you'll see it again here shortly in video format, or in demo format. I'm going to demo it out, but you'll have to calculate those RF factors. Okay. The microanatomy, the number of stomata, okay, so you're going to want to concentrate on that. The CO2 consumption, and you're going to have to fill out this answer sheet, drawing the microanatomy, answering these questions, and that will get you where you need to go for photosynthesis. Make sure that you're filling out table 3 when it comes to carbon dioxide consumption. Okay, so we're going to jump over to the videos of me doing this lab, and then you should be able to answer these questions. Hi all, Dr. Clark here again. Today I am going to demo out the photosynthesis lab. Okay, so the first procedure in the photosynthesis lab is looking at plant pigments and comparing two types of plant pigments and seeing, basically what the plant pigments are that are in that plant. So what's their RF factor, or what's the distance that the plant pigments travel through chromatography paper. Okay, so I have the system set up already. I've already done one just for the convenience of videotaping time. Okay, so in this mortar and pestle, I've already extracted the different pigments from a money plant. Okay, so a leaf of a money plant. Okay, and I've already placed it on this chromatography paper in that center. So now I'm going to do the same for this geranium leaf. And so I'm going to demo that out so you can kind of see how that works. Hopefully you'll be able to see this. So inside the mortar and pestle, I have some just some sand. I'm going to take this geranium leaf, rip it into a few pieces, add roughly 5 mils of acetone, and then I'm just going to give it a smash and a crush, and I'm going to try to get those pigments to come out of these leaves. So it doesn't take too much effort, but you'll see that eventually I'll get a fair amount of pigments that are mixed in that acetone, and then we can apply it to the chromatography paper. Okay, so there you can see, hopefully, I have quite a bit of pigments from the plant, and now I'm just going to take a capillary tube and get some of that pigment to come up. Maybe that capillary tube's kind of getting clogged, and then what I'm going to do is, hopefully you can see this, is on my line that I've drawn on the chromatography paper 2 centimeters up from the bottom, I'm going to just place a center dot, and I'm just going to keep on placing dots. Maybe I might need to get a new capillary tube. There we go. And then, so I'm just going to place center dots. And once it's saturated, you're going to have to let it dry for a little bit. Acetone dries really fast, so you'd have to just let it dry and then keep applying dots to that center line. And the idea is that you're laying down these pigment molecules, and as you lay down those pigment molecules, we're going to run them through chromatography paper, which will separate them based on size and how far they're going to travel through the solution. So we want to make a pretty good band of pigments, because the more pigment that you lay down on that one band, the better chance that you'll see nice color bands, nice, levels of chlorophyll A and B and xenophils and carotenoids along that band. So that's probably pretty good. It's pretty dark right there. Okay, I'm going to give it a second to let it dry out. Okay. And then I'm going to show you, so inside these test tubes, this is plant A, okay, but inside these there's a chromatography solution, okay, and that chromatography solution is just 9 parts petroleum ether and 1 part acetone. Okay, I've already mixed it up, thrown it in there, okay, and now we're just going to place our chromatography sheets inside that test tube. You need to make sure that you have a cork or a lid, because petroleum ether and acetone, they evaporate really fast and you won't have much solution left. So I'm just going to toss it in there. Alright, try to kind of straighten it out a little bit. Well, loosely put the cork on, and then you should be able to see that that pigment is starting to move off that line and travel with the solvent up the chromatography paper. As it moves up, molecules that are different sizes will come out in solution at different, or along the paper at different times, and then we should be able to separate chlorophyll A, B, xenophils, and carotenoids. Okay, they're going to be different colors. Chlorophyll B is going to be kind of a lighter green. Chlorophyll A is going to be kind of a sea blue. Xenophils are yellows, and carotenoids or keratins are orange and red. Okay? And as that progresses, we're going to eventually see that it'll make close to the top. When it gets closer to the top, I'm going to stop it. You want to make sure that you stop it before the solvent reaches the top of the paper. Okay? And so I'm going to stop the video. This takes a little bit of time, and then I'll show you the results in a little bit. Okay, so the chromatography paper is dry now. It's out of solution. It ran for about 10 minutes or so in order to have the solvent go about a millimeter or so, well, about a centimeter or so from the top of the paper, and that's when I called it quits. and so I can show you the results. Now hopefully it's going to pick up on the camera. I don't know, some of the lines are really faint, but I have a little mark next to the line because I took a measurement of the distance that the solute traveled, also the distance that the solvent traveled. So if you look here, you can see this is species A, or plant A, that's the money plant. You can see there that the first peak, that light green peak is, the first peak, and then you have kind of a sea green, then you have a yellow, and then you'll have a really faint yellow line right below the solvent line. The solvent line is the last line on that paper. And then B, it's kind of curling on me, but same kind of deal, same pigment colors basically. Just you can see the travel patterns very different. Okay, the chlorophylls didn't travel as far. Okay, the xenophil traveled much further than the xenophil in A. Okay, so there's a lot more distance. And then there's a better carotenoid look at, or a better carotenoid pigment line than I saw in the money plant. So it looks like the money plant has, better chlorophyll pigments, maybe even more chlorophyll pigments based on this. That's hard to tell, but at least a better pattern than the geranium, but the geranium seems to have a longer peak on both the xenophils and the carotenoids. Now, I have the data for you here. So, plant species A, okay, the money plant has a solvent distance of 85 millimeters. The keratin or carotenoid distance is 63. Xenophil is 57. Chlorophyll A is 55. Chlorophyll B is 41. Plant species B, solvent, it's about the same, 83. The keratin is 58. Xenophil is 50. Chlorophyll A is 35. Chlorophyll B is 24. So the distance traveled is a little bit different. Okay, that tells you a little bit about the molecules in general. These molecules could be bigger as long as we did it correctly and about the same amount of pigment was laid down. Then plant species B might have heavier molecules. Maybe not, but least we can do is calculate the RF factor. It tells you that, it gives you an example of the distance traveled. And different RF factors will allow you to differentiate between different plant groups. And so you can tell these two different plants have different RF factors. So first for you guys is to calculate out that RF factor. The next thing that I'm going to do is demo the leaf microanatomy. Okay, and so that video is coming. That video will be kind of interesting video. It'll be a picture of me looking through a microscope, but then I hopefully will get up a live video of the microscope, me scrolling through and talking about the different pieces in that leaf microanatomy. Okay. Well, all right. So now we're going to look at the microanatomy of a leaf. Okay, so this is the setup I'm going to work with. So you can see I have a compound light microscope sitting here. Okay. I have a leaf epidermis slide on it. Epidermis being the top layer and the bottom layer. This one happens to be the bottom layer of the leaf because we're going to be looking at stomata. Okay. And so the best way for me to show you this is by hooking the compound light microscope up to a computer. Okay, so I'm going to show you the images that I see, through the computer screen. So hopefully it'll be clear enough that you can check it out. Right now it's blurry because I haven't focused it, but I'm going to focus it under 4x or 40 times your eyesight. And again, this is the epidermis. Okay, and You can start to see it come into focus there. These little cells right here, these are the stomata. So that's the opening to the cell, or opening to the leaf. Okay, this is where carbon dioxide would enter, oxygen will leave, and water will also escape through transpiration. Okay, so that opening is surrounded by guard cells. Okay, so let's zoom in a little bit more. So let's go 100 times your eyesight. Okay, so that's pretty good there. And you can see guard cells. You can see the nucleus in each one of those. You can also start to see these little structures, other organelles. Majority of this is chloroplast. Even though it's not green, that's because this slide has been dyed blue, so it's a little bit easier to see the stomata and see the guard cells in the opening. Okay. Let's go to 400 times your eyesight and look at kind of an individual stomata. Let me see if I can get it on the slide and focus, maybe. Okay, there's one there. And you can see, now you can see the chloroplast. You can see the nucleus. This is a guard cell, and this is a guard cell, and this is the stoma, the opening to the leaf. Now again, these guard cells, their purpose is really to regulate when this is open and closed. Okay, they do so by osmosis and regulating the size of the cell, and how much water these cells have inside them will determine whether or not this is open or closed. Okay, and some other properties that are going on. Okay. Now, since they have chloroplasts, they have chlorophyll, and these are also sites of photosynthesis. So the guard cells, the stoma, where the openings are, can also do photosynthesis, and they do. So this is a good site of carbohydrate building or carbohydrate construction also in a cell. Okay, so that's stomata, the guard cells. Now we're going to look at kind of a big picture on what does the leaf itself look like in cross-section. So what I have here is just a simple leaf, and I'm going to show you the cross-section of that leaf and explain to you the different microanatomy that leaf has. Okay, so there you can start to see it come into focus. Okay. And we'll zoom in on this, but real quick, okay, this is just a cross-section of the leaf. You can see a big xylem, phloem, kind of vascular region there, and there's some smaller ones throughout. Okay, but this top faint pink line is the cuticle layer, sometimes called the epicuticle layer because it occurs next to the epidermis. Okay, that's the waterproofing layer. Then these clear cells are the epidermis. Then you have the palisades perichinum. So that's these kind of column-looking cells that are kind of linked together. Then you have what we call the spongy perichinum. Okay, and they get the name because they have these open regions where water can be stored, oxygen, carbon dioxide can come into here, and it can be passed into the regions of the cell that need to utilize those. And then it's hard to see, but at each one of these kind of what you could say is air sacs or open areas, you're going to have some stomata. You're going to have these guard cells. Okay? There's one there. There's some there. Some there. And we'll zoom in and look at these in just a second. Okay? So now going from 40 times your eyesight to 100 times your eyesight. Okay. Now you can see quite a bit more detail. Okay. And so now the cuticle layer is much easier to see, that pink layer. the epidermis, okay, palisades perichynum, spongy perichynum, okay, xylem phloem. Okay, this is how water and nutrients get into this region. Okay, so that's how material gets carried to the plant leaf, is in these vesicles. And then you have guard cells, these stomata openings. There's one there, there's one there, and there's another one down there. Okay, so we're going to zoom in more, check them out a little more. So 400 times your eyesight. So now you see just the spongy perichynum. Okay, and then I'm just going to kind of move it over a little bit so you can see one of those guard cell stomata openings right here. Okay, not as easy to see as looking at the bottom of a leaf, but nonetheless this is an opening, a pore to this cavity here. Okay. And then if we move up the leaf, so from the spongy into the palisades perichynum, through the palisades perichynum, okay, to the epidermis, and then you can see that cuticle layer. Okay. So that's the microanatomy of a typical plant leaf. Okay. And next we're going to bring you ways that you can analyze the amount of stomata that a plant has. Well, Dr. Clark here again. Alright, so now we're going to switch gears just a little bit, and I'm going to show you how you can look at the stomata of plants, okay, and you can get an estimate of how many stomata openings different plant species have. So you can see this Sabrina plant here. I'm going to just take some nail polish, clear nail polish, okay, and I'm just going to paint a strip of nail polish on the bottom of a leaf. So if you can see this leaf right here, I'm just going to take and run a strip, okay, and let that dry. What that's going to do is make a What that's going to do is make an imprint of the stomata that are underneath that leaf. So after it dries, then I just take some general Scotch tape and stick it on top, and then gently peel that off. And here's what you're going to get when you do that. So there is the camera, and I have a stomata print from a money plant. So you can see there the stomata. And so what you need to do is count how many stomata there are. Let me actually increase the size of the screen a little bit. You're going to count how many stomata there are in a given field of view. So each one of these stomata imprints are 1 centimeter by 1 centimeter. Okay, so a centimeter squared. Now you're going to need to figure out how many stomata there are for a centimeter squared. And what I'm going to give you is this picture here is giving you how many stomata there are for 2 millimeters squared. So you're going to have to do a little bit of math and convert 2 millimeters squared. Okay, you're going to have to times that out to 1 centimeter squared. And you'll have to count the stomata. So what are you counting? You're counting these guys. So you can see there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45, 46, 47. Roughly 47. If you get a different count, great. Use your count. But 47 for a 2 millimeter squared area. Okay? So you're going to then take that and you're going to use that information to see which plant has more stomata openings. Okay? So that's the money plant, species A. And now I'm going to throw on the geranium, species B, and same kind of region, same area, same magnification, and now you can see the stomata here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 for 2 millimeter squared area. Okay? Doesn't take rocket science to know which one has more, but you also need to make that mathematical conversion to tell me how many stomata they would have for a 1 centimeter squared area. Alright, so this is 2 millimeter square area. You're going to have to convert that to 1 centimeter squared area. Alright, so that is the stomata imprint, okay? And it's pretty easy to do, easy to see, and can give you a little information about the environment that an organism lives in. Hi, okay, so now we are going to demo out the uptake of carbon dioxide by photosynthetic plants. So what I'm going to do is I'm using an indicator dye here, which is called phenol red, and it's a combination of phenolphthalein and some other solutions, but regardless, it's an indicator of an acid. So phenol red's an indicator of an acid. So to solution A, or beaker A, I'm going to add a piece of Elodia, which is a species of aquatic plant, and to B, I'm going to add a piece of Elodia, which is an aquatic plant, and to beaker C, I'm not going to add anything. It's going to be my control. Now, beaker B, I'm going to wrap in aluminum foil so no light can penetrate. So I'm going to show you what I did to beaker A and B to make it so they are loaded with carbon dioxide. So what I'm going to do is I'm going to take the beaker and I'm going to blow in it, producing carbon dioxide, which will then produce carbonic acid in here, and will change the pH of this to be more acidic. When it becomes more acidic, phenolphthalein, phenol red, will change from pink to yellow. And then through the experiment, we're going to see if plants can change the color from yellow back to pink, meaning that they're taking carbon dioxide out of the water. So I'm going to give this a little bit of carbon dioxide right now. Okay, and so those beakers are all about the same yellowness, okay? But just to make sure that we're on the same playing field, okay, I'm now going to get an initial pH value. Okay, so I'm going to put the camera here, and I'm going to show you how to test the pH with pH probe. Alright, so the pH probe's coming on. I'm going to take the probe out of fluid, okay, and then I'm just going to dip it into A. I'm going to let it rest there for a little bit until it evens out. You can see that, well, maybe you can see that the values are going down. Eventually it'll stable out. So that took a little bit of time to get the pHs, but I also wanted to make sure that you understand that there's no smoke and mirrors going on here. So the pH of water is 8.15. The pH of phenol red in water is, well, according to the meter, 8.32, but it's been jumping around from about 8.30 to 8.35. So 8.30. Beaker 1 starting out 7.38. Beaker 2 7.35. Beaker 3 7.39. Okay, so I'm going to call Beaker 1 A, Beaker 2 B, and Beaker 3 C. Okay, I'm back and I got the plants in. So this is Elodia. And so you can see that green on that plant. That means that there's chlorophyll in there and it should be able to do photosynthesis. And so I got about 10 centimeters of plant in both Beaker A, okay, and Beaker B. And then Beaker C is empty. There's nothing in there. Okay, so there should be no photosynthesis going on in Beaker C. So now what I'm going to do with Beaker B is just wrap it up, okay, and then slide them all and then in line by the light, and then it's a waiting game. So we're going to wait an hour, and then we'll come back and we'll check the pH and see if we get a change in pH or if we get a change in color. Okay, so I'm back and it's been an hour, and now we're going to take measurement of the pH for the different containers. So I already did the control container, and now the control container, so the water, and now so I did the water, I already did the water container, and I already did Elodia in the light. So you can see, most of you probably can see that it's starting to turn back to pink, okay? And this is Elodia in the dark. So now I'm going to look at what Elodia in the dark is and give it a chance to see that. So right now we have Elodia in the light, or tube A. It went from 7.38 to 7.72. Remember, 8.30 is the original starting material. Okay. Elodia's, or the control also changed, 7.39 to 7.64. So to explain that, you can imagine that as time goes on underneath a hot light, that you're going to get evaporation. And so water molecules are going to be evaporating, but also carbon dioxide is going to be leaving, and oxygen is going to be coming in, and vice versa. So there's probably some mixing going on, and that's probably associated with that 7.64. And if we look here, Elodia in the dark, it looks like it's about 7.60, so pretty close to the same mark as the control. Okay, so if you look at that 7.35 to 7.60 or 7.39 to 7.64, it's about the exact same distance. Technically it is. This one's a little bit more, a little bit different, 7.38 to 7.72. Okay, we're going to let it sit for 2 hours to see if we can get a more drastic change. Okay? So I'll bring it back in 2 hours. I'll also calculate what is going on in water and just phenol red. Okay, so I'll bring you back 2 hours. It's been 2 hours now since we started the experiment. It's pretty noticeable now color-wise. So here is the control sample. You can see here is Elodia or Elodia in the dark. Okay, those two are pretty much the same color. And there's Elodia in the light. You can see that the pink is starting to come back. I already have the two values for Elodia in the dark and Elodia in the control sample. They're both at 7.66 for a pH. So now we're going to check out the pH of Elodia in the light to see if photosynthesis was occurring. And hopefully it'll kick over relatively quick so I'm not wasting a huge amount of time. But just from the color, you should be able to see that it's coming back. It's removing carbon dioxide out of the water, and the pink is coming back. The color's coming back into the system. So it's starting to slow down now. 7.98, 97, 7.96. Okay, we're going to call it a 7.96. If we switch it over real quick, see what the original sample was, maybe. Okay, so this is still in the, 8.5. Remember, it stopped at 8.32. I'm guessing it's going to be about 8.32 given the rate that it's going down. So I'm going to stop the video there, and you can tell here are the numbers. These are basically the same, 16, 17. I already did this one, and then we'll just call this the same as the last time. As you would expect, you shouldn't see much of a difference between the two. You do see that my water is a little pink because I carried over some of the dye from the probe. But nonetheless, photosynthesis, so tube number A or tube A, went from 7.38 to 7.96 in 2 hours. B went 7.35, 76.66. C 7.39, 7.66. Now, it's quite possible that B had a little bit of photosynthesis going on. Now, remember that the portion of the photosynthesis that would consume carbon dioxide is the Calvin cycle. Okay? And so the Calvin cycle can happen at night, and it does so in C4 plants. Okay? C3 plants, often it's just occurring during the day. But in C4 plants, remember, they can bring in carbon dioxide and they can store it as an acid and hold on to it. And C3 and Elodia is both a C3 and a C4 plant. Okay? It has the enzymes for both, so it is quite possible that it was doing photosynthesis at least a little bit while it was in the dark. Okay. All right. So that should be enough information that you can complete the lab.